to the need of all the people who listen to us for more information. And so we're so pleased today to have with us the world's
expert
on rhizophagy, Dr. James White, from right here near me. I'm 1 hour from Rutgers University, so he's from Rutgers University. And so what I'd really like to do is turn the stage over to Dr. White, who will introduce us to some of the basic facts about rhizophagy.
Can I say a couple housekeeping items first? This is Laura from microBIOMETER. We're hosting this as a Zoom call, not a webinar, so it's a little clunky, and I appreciate everyone's patience. We're also recording the entire presentation, and it will be on the microBIOMETER YouTube site. I encourage anyone in the audience to mute themselves, to put on speaker view, which is the top right view option, and to use the chat. If there's any issues, please reach out to me or send an email to info@microBIOMETER. Okay, so now we'll turn it over to Dr. White. Thank you.
Okay, thank you so much. I'm very happy to be here to be able to speak with you this afternoon. What I'm going to try to convince you of is that plants are cultivating microbes the way that we cultivate plants. Essentially, plants are farming microbes is how I like to put it. And you know, I'll describe details of the Rhizophagy cycle as we currently understand it, and I think you'll see that plants seem to be in charge of this cultivation process of soil microbes. I'm going to try to share the screen now. See if it works. Okay, I think it did. And okay, so first, what are endophytes? Endophytes are microbes. It could be any kind of microbe that goes into the tissues of a plant. And that could be going into plant roots, into plant seeds, into the stems, into the leaves. Endophytic microbes can be fungi, and they can be bacteria, and in some cases they can be algae. So plants have these microbes that go in them, and usually it's not one microbe. It's a plurality of microbes. It really is a small community that goes into plants. And you may have heard of endophytes in grasses, and there are certain grasses that will have these fungi that go in them, and they will go into the leaf tissues, particularly of the plant. Those are endophytes, and this actually is an image that shows these hyphae of the endophyte in a grass, and you can actually see the big cells in the background there, and those are the parenchyma cells inside the grass plant. And then you can see the hypha, this blue stained structure. Okay, that's the endophytic fungus. Fungus actually goes by the name of epichloi. You don't have to remember that. It's a complicated spelling, but it's the common endophyte that's in grasses. But of course, all plants have endophytes. Every single plant has endophyte. Grasses have these fungal endophytes, but they have other fungal endophytes, and they have other bacteria. Every plant has a community of endophytes in it. There are no azenic plants. There are no plants without microbes. It's very difficult to remove microbes from plants. It takes a concerted effort. I have a graduate student, April Michi, who is beginning to work on hemp endophytes and trying to understand what endophytes are there and how they benefit and affect hemp. And you know, if you look at the hemp seed, for example, you can take that and germinate it in an azenic situation, that is on sterile agar or sterile medium, so that new microbes can't get on it. What you see is this actually is a root hair, and of that hemp seedling. And you can see these little brown dots in there. This is stained so that we can actually visualize the bacteria. Otherwise, we wouldn't be able to see these bacteria. This would be totally invisible. And what you can see is that these little spherical structures are actually outside the membrane of that root hair. I mean, you see the wall out here, the thick thing, and then you see the cell, the root cell itself is kind of constricted in here, kind of collapsing a little bit, and you see those microbes on the surface of it. Okay, those are all bacterial endophytes in that plant. All plants will have this. They'll have a community that actually go inside the cells of the root. Here's another plant. This is a plant, a non-cultivated plant. This is a cactus called the Kadushi Cactus on the desert island of Bonaire in the Dutch Antilles. And these things are huge, these cacti. And they produce up high little fruits like punchia fruits. And you take those fruits, cut them open, pull the seeds out so that you don't get soil or anything on it, and then take those seeds and germinate those seeds again under azenic conditions. You get a seedling looks like this, and you can see the roots here, and you see all those root hairs coming out. Now this one, those roots are orange brownish in color, and the root hairs all over it because we stained it for reactive oxygen with the stain we call DAB, but it stains hydrogen peroxide. And when we use that stain, we can actually see the bacteria inside the plant because they will elicit reactive oxygen from the plant, and so they're visualizable. This is a root hair of that cactus, and you can see the arrows indicating these little bacteria, little pairs. They're in pairs where they just divided of bacteria inside those root hairs. So all the root cells will get these microbes in them. Turns out shoots also get the microbes, but the microbes in the shoots are not long-lived. They will break down eventually, but the ones in the roots will actually participate in the Rhizophagy cycle, and they'll go into the soil and back and actually acquire nutrients. And I'll show you how that process works. Where do these microbes come from? These are endophytes, but they are soil microbes. The plants actually, all of these microbes are really fished out of the soil by the plant. And the beneficial microbes, the plants typically will take and put on their seeds so that the seeds now can carry them. And when the seeds germinate, the seedlings will have these microbes and they'll be able to grow perfectly and to be defended by these endophytic microbes. But other microbes, well, some are fished from the soil and others really come with the seed. Now, the first source, even for those that come with the seed, is the soil. So every one of these can go back into the soil and function in the soil. And that was their initial source, was the soil. And that's the importance of having a healthy soil with a diverse microbial community. Okay, this internalization of microbes into roots was observed by an Australian group of investigators in Queensland, Australia, and this is one of those investigators, Channy Pong Fu, and they saw that bacteria and yeast were going into roots, and they actually did an experiment using tomato and Arabidopsis, where they took their yeast and a bacterium, E. coli, and they labeled it with green fluorescent protein. They labeled it or tagged it so that it would then fluoresce with this green fluorescent protein, and they could track it in the plant. Then they took those microbes and they fed them to tomato plants and Arabidopsis plants, and then they tracked them into the roots of the plant. And you can see some of their images here. If you see B up here, you can see all these little dots in there. Those are actually the bacteria that are inside the root cells. And you can see a little hair thing coming out. That's the bacteria inside those hairs. And you see these, number D here, you see these elongate structures. Those are the root hairs. And there are lots of bacteria inside those hairs. If you look at E over here, you can see a hair and you see yeasts now, these little green dots. Okay, so those are all fluorescing. So they know that the bacterium that they labeled and then fed to these plants went in. And what they discovered was that in time, in 3 or 4 or 5 days, those bacteria disappeared from in those roots. So their hypothesis, their conclusion was that the plants are actually consuming these microbes. They're degrading these microbes and they're obtaining nutrients from it. And they later called this process rhizophagy. Rhizophagia. Paper, actually a chapter that comes a year or two later, they named it rhizophagy, rhizo for root, phagy for eating, to indicate that the roots are eating these microbes. What we have discovered, okay, their discovery was about 2010. And since then, we've been working on this process. And what we have found is that it actually is a cyclic process. And that is that microbes from the soil or from the seed coat will actually be attracted to the root tips of plants. And that's because at the root tip, plants are secreting exudates, and those are sugars, organic acids, and sometimes amino acids. And the microbes will be attracted to there, and then they'll be internalized into the surface layer of cells just behind this meristem. They'll be internalized into those cells. And then once they're internalized, the root cell will hit those microbes with superoxide or reactive oxygen. And that will strip the cell walls off of the microbes. It will also extract nutrients from the microbes. And then what happens is those microbes, the surviving microbes, are replicated actually inside those root cells. And then the surviving ones will be ejected. They'll trigger root hair formation and they'll be ejected out into the soil where they'll reform their cell walls, and then they'll go out into the soil and acquire more nutrients. Okay, they're ejected from the tips of those elongating root hairs. And as those root hairs elongate, they eject those microbes. When we take all the microbes away from plants, we don't have any root hairs forming. So these microbes are important for triggering and continuing the formation of root hair, of the root hairs and root hair elongation. When there's no more microbes in the root hairs, elongation of the root hairs stop. No more elongation. Okay, so this actually is a root of Phragmites australis, this invasive grass that's all over North America. And this is the root tip here. And you can see this cloud of blue out here. Those are the microbes in that exudate zone. And you can see it's kind of diffuse outside, and you see more and more higher concentration of those microbes as they get close to that root. And what happens at that interface, those bacteria are taken into the root cells. We don't know exactly how they're taken in. We don't know if it's some kind of an endocytosis phenomenon, or if the microbes themselves are degrading their way into the cells, or if they're just pressed actually into the cells themselves. These cell walls are very thin, and around after they form, after they divide, they're not yet hardened. So it's possible to easily go through that cell wall. Once they harden, it's hard to get in. But right in this zone, the cell walls are soft, and so these microbes can get in. And so they do go in. We know they go in because we can see them in those root cells in this zone. Okay, this actually shows, again, the Phragmites plant. This is a root cell, an epidermal root cell. And you can see where these black arrows are, and all around these other areas, these little spherical brown structures, those are the microbes that are inside these root cells. And these microbes are, at this point, are protoplasts. They're naked protoplasts. These were actually pseudomonads when we first put them on the plant, and they were rods. Okay, but what happened when the plant hit them with superoxide? It oxidized off the cell wall, and they become these spherical structures. And these spherical structures divide inside the plant. And if you look at the lower arrow, you can see a big cell that's not staining very much internally. And then you see the smaller one, the smallest, stains blue. That blue staining is from aniline blue, and that's staining the protein in that cell. And so what's happening is these older cells are actually being broken down, they're being degraded, and the protein is being extracted from them. But the plant will actually replicate these microbes inside these cells, in addition to break some of them down and extract nutrients from them. And the superoxide is expected to make the membranes around these protoplasts, microprotoplasts, leaky. That's one of the effects of superoxide. This is a diagram of a plant cell, of a root cell, plant root cell. And the brown here actually is the cell wall, or brown, the gray. It's gray on the outside, the cell wall. And then the white is actually the cytoplasm of that root cell. The microbes don't actually go into the cytoplasm of the root, at least not in the Rhizophagy cycle. In the Rhizophagy cycle, they will be limited to just underneath the cell wall, in this space between the cell wall and the plasma membrane of the cytoplasm of that root cell. Okay, so they go in this little space just underneath the cell wall, and you see bacteria there. And it's into that space that the plant secretes superoxide. So the plant is able to detect these microbes there, and it will secrete superoxide right in that zone, right around those bacteria. So it's focused on the bacteria. So the plant is detecting these microbes, and it's putting superoxide there. Now, there's another phenomenon that I'll explain later, but basically the plant is also moving these microbes around in the process we call cyclosis, and you'll see that. But it takes the cytoplasm and moves them around and around. And what that does is it moves these microbes around. So it's jostling these microbes around. And I'll show you images of that, movies of that really. These microbes, internal in the root cells, are critical for proper development of plants. And basically, two effects are notable or really profound. And one is that the microbes in the roots will trigger the gravitropic response. And they will the gravitropic response for plant roots is when they will actually grow downward like this into the soil. Okay, if we remove all the microbes away, plants lose that gravitropic response. And what happens is the roots will then grow either up into the air in some cases or flat on the surface. Very rarely then do they actually grow downward. Okay, and the other effect is that if we remove all the microbes from a seed or seedling, then they no longer form root hairs on the roots, and the roots become totally hairless. If we put those microbes back on, then you see hair formation again, and you see gravitropic response. Okay, so there seems to be plants actually seem to have evolved to require these microbes in their tissues. And this actually is a Bermuda grass seedling. And this is in this experiment, what we did is we removed all the microbes onto Bermuda grass by surface sterilizing very harshly, rigorous surface sterilization. And many grasses actually have all the microbes vectoring on the surface, so we can do that. And when I'm talking about rigorous sterilization, I'm talking about like 50 minutes to an hour using like 50% Clorox, which is 3 or 4% sodium hypochlorite, with constant shaking. So this is very, very vigorous to get all those microbes off. And you can see the root here. This is stained with our DAB stain for hydrogen peroxide. But you can see the root there. You see the tip out there. You can see the root axis here. There's no hairs there. And this is a bit older to the right, this image. And you see there's no hairs there either. Without those bacteria, inside the root cells, there's no hair development. Okay, here's where we took one of our pseudomonads, one of our bacteria, pseudomonad, from Phragmites, and then we put it on this plant where we removed all the microbes off of our Bermuda grass, and then we put it on some seedlings. And then what you see is this: immediately you see hair formation. You see the root hairs developing, and you also see it's staining. It's not that staining is the reactive oxygen. So when those microbes are there, the plant then reacts to them and secretes reactive oxygen. But those microbes are in those hairs, stimulating hair development. And I'll show you more about that. This is an older part of the root. And you can see this is again stained for the reactive, the hydrogen peroxide stain, the DAB, the DAB stain. And you can see all the brown dots. Those are all the bacteria. And if you look at the axis of this root, the main axis, you can see little brown dots there. All of these cells are filled with these microbes. These plant roots become full with these microbes. Every root cell. Now it only goes so deep. It typically is 1 or 2 cell layers on the outside where these microbes are for the Rhizophagy cycle, but that's where it's important that they are. This actually is a root hair from that not Phragmites, but from the Bermuda grass, and you can see the hair. You can see inside the hair all these little brown dots. Those are all the protoplasts of that Pseudomonas, of that bacterium. These root hairs become engorged with these bacteria. They become very filled with these bacteria. And one of the things that we've been studying, I don't want to dwell on it, but it is something important, is why is there a linkage between hair elongation, root hair elongation, and presence of the microbes there. And we've been trying to figure that out. And we've developed a couple of main hypotheses. One is that it is ethylene. The other is that the microbes are secreting ethylene. That's also a plant hormone, and that triggers elongation. And the other hypothesis is that the microbes are secreting nitric oxide, and that also is a plant hormone that's known to be involved in root hair elongation. And so those are our 2 main hypotheses. Lately, we've done an experiment. My student, Ivy Chang, did an experiment to try to evaluate which it is. And here, actually, this was with the POA bluegrass, but you can see this is with no microbes. This is the root, and you can see there's no hairs there. Okay, and then she put microbes back on. And here you can see the root where she put the microbes on, and you can see the hairs again. And you can see the little dots at the tips of those hairs, those little heads there. Those are filled, those are actually the microbes that are coming out of those hairs. Okay, so this is the system that she used. And the hypothesis that we're favoring now is that these microbes are secreting ethylene. And that ethylene, and what happens is the microbes accumulate in the tips of the hair, and this is a picture of that. And it's stained brown here, and all those little dots are the bacteria in that hair tip. And their presence there in a big group, secreting ethylene, causes growth in that hair. And so elongation happens, and root hairs then elongate. And just to show you the extent to which we are trying to figure this out, here's a chemical reaction, actually, to what we think is happening in those hairs. And basically, we think the microbes are using this enzyme called microbial ethylene synthase, and that enzyme utilizes arginine, and it will then produce ethylene. Ethylene, being a plant hormone and produced by these microbes, will then trigger hair elongation in the root hair, but it will also trigger superoxide production, and that's where the superoxide is secreted onto the microbes. That the superoxide and the ethylene will combine, and then it would produce hydrogen peroxide, and that's where our stain, the DAB, is picking up the hydrogen peroxide. We can also stain for superoxide, and we can see the superoxide in there. And recently we have stained for ethylene, and we can see ethylene in there. One of the tests that we did that suggests that ethylene is the signal, the elongation signal from the microbe is we used an inhibitor of this enzyme, of ethylene synthase. And that inhibitor is an arginine analog called L-name. And so when we put L-name in there, it gunks up this enzyme because it doesn't work like arginine. It stopped the whole process. And what we got no elongation and we got no superoxide formed in that case. So we still have to test it, but we're leaning towards ethylene as the signal molecule. Okay, this is actually a tomato root from a tomato seedling. And it shows the area where microbes enter the root. And this actually is this is the meristem here. That's where cell divisions occur in the root. And then this kind of purple orange color, this is actually where the microbes are entering. This is stained for ethylene. So where they're entering is where we're seeing some ethylene. We're also seeing it here at the root cap. We also know that the microbes are entering the cells at that root cap. So these are the two zones where we're seeing ethylene. Okay, this is a root cap cell. You can see the microbes inside that, and you can see the kind of red, orange, purple, blue to purple color of those cells where we think ethylene is produced. These are the bacteria outside the cells, and you can see also the blue that should be from the ethylene. That's how we interpret that. This is in the root hair itself. You can see all the spherical structures. Those are all the microbes in those hairs, and that's standing for ethylene. So they're producing ethylene, it looks like. Okay, so this is one of the plants that we examined to study the Rhizophagy cycle. This is a little sedge, and the sedge grows in the kind of the limestone formations on the island of Bonaire. And this sedge grows on these rocks in little pits. It doesn't have much soil, maybe a little sand, but what it does have is a lot of these microbes. And we'll see we did experiments with these plants, and this is kind of cool. This is a root hair of the sedge below, and this root hair below is stained with that DAB so that we can see the hydrogen peroxide around the microbe. And you can see the arrows here, these little black arrows, and you can see pointing at these little structures. Those are the microbes. You can see the brown around them where hydrogen peroxide is. That's from the superoxide that was converted into hydrogen peroxide. Okay, so you can actually see the red in there, and you can see the blue for the microbe. And you can also tell these microbes are in the periphery. They're not in the cell per se, but they're in the apoplasmic or the paraplasmic space. They're just underneath the cell wall. And above, this is a living root hair of that sedge. And you can see the shadows that move around. This is the cyclosis that I'm talking about. So the root hairs are processing these microbes. They're moving them around, cycling them around and around. And we think that's important for a couple of reasons. One reason is that because the plant moves those microbes around, it creates a situation where there's maximum nutrient flow between the microbe and the plant cell. If that were static, just sitting there, what would happen is you would get gradients that would build up, and that would slow down nutrient flow. So this movement breaks down those gradients and makes nutrients flow rapidly from between the two, because there is nutrient flow between the two. The plant is producing photosynthate, and it's feeding these microbes, and the microbes their nutrients are being extracted from them. And so there's need for nutrient exchange there. The other effect of that movement, of that cyclosis, is that the microbes are broken up by the plant. The plant breaks them up into many, many copies. It essentially will clone these microbes so that whereas a few microbes, a few bacteria or yeasts go into the root cells, many will come out of the hairs. It's replicating them. This is that sedge again, and it shows kind of the whole thing. And you can see the cyclosis, you can see the movement, the little shadows going around, and you can see at the tip, all these little white things jostling around the tip. Those are the microbes that are accumulating in the tip of that hair. That's how this works. Hairs move the microbes to the tip, and they concentrate them there, and every now and then we'll move one and catch it and move it back. So you have that, but you have concentration right there in the tip. This shows ejection, and this ejection occurs periodically. The microbes, what happens is there begins to be a swelling at the base of these hairs, and that swelling propagates to the tip, and you can see it at 1, you see it at the base, but at 2, you can see it a bit further up, and you see it swelling up and pushing those microbes out. And number 3, you can actually see the hair. This is tomato, by the way. You can see the hair with, at the tip of the hair, you can see 3 little pores, if you really look at it, 3 little pores right there where those microbes are being squirt out through. They're being pushed out through. We don't know what initiates this. We do know that it likely has to do with potassium, because we can put potassium in the medium and we can increase this process. So we think it's potassium loading into this, the vacuole that is in this hair. And then the water then goes into follows the potassium and makes this swell and pushes out those microbes. But that all has to be proven. So, but what happens once these microbes are ejected is they will rebuild their walls, go out into the soil, rebuild flagella if they have flagella, go out and acquire new nutrients. Okay, what happens to plants without the Rhizophagy cycle is this, and this is rice. It's an experiment with rice, and what we did in this particular experiment, we removed all the microbes off rice, and that process involved Clorox, and for about an hour, surface sterilization, similar to the other grass, and treating with antibiotics. And then we put microbes, put a bacterium back on. And this E plus here, this is endophyte plus, but it was a bacterial endophyte that we put on. And you can see, and this one to the right, E minus, we didn't put any endophyte on that. That is an azenic seedling. And okay, you can see the difference. With endophyte, it grew much better. And this is a week or so later. And you can see with the endophyte, you can see the seedling is much bigger. These are the same magnification. The root's about the same size, but you can see the soil is adhering to this root, these roots over here, and not over here where we removed the microbes. The presence of the microbe creates a very close association then with the roots. The soil and the roots associate closer than they do with no microbes. And that's apparently critical for nutrients. It makes a big difference to the plant. So the microbes really improve interaction. One effect is interaction with the soil. Okay, the Rhizophagy cycle, we are convinced that it's a nutritional process for a number of reasons. One is because of experiments that we've done. And one type of experiment is this isotope tracking experiment. And basically in this experiment, what we did is we labeled proteins with isotopic nitrogen, this 15N nitrogen. Instead of regular nitrogen in the protein, we put isotopic, this 15N type nitrogen. And then we incorporated it into this medium, this agar. And you see this gelatin here that has that labeled protein in it. And then what we did is we put fescue grass with microbes on one or some of the plates, like you see here, and then without microbes over here on other plates. And you see here, and one difference is you can see where we had the microbes. You see more reactive oxygen. You can also kind of tell these seedlings are a bit bigger than without the microbes. And okay, that's just how they look. But really, what makes the difference is actually measuring that nitrogen. So then we went in and we took these shoots off, and then we analyzed them for how much nitrogen, how much isotopic nitrogen went into each one. And here we have, without the microbes here, this middle one, this middle bar, and you can see it goes up to, a certain point. And then if you look to the left, here's with this big bar here, this is with the bacteria present. You can see it went up higher. That's actually about 30% higher than without the microbes. So we interpret this to indicate that our microbe presence in the Rhizophagy cycle is worth about 30% of the nitrogen that those seedlings, those fescue grass seedlings were getting. Okay, we've done other experiments where when winter wheat and also with other plants. But with the winter wheat, we had a visiting scientist, and this him who did the work with wheat seedlings. But what he did was isolate some of the microbes off of wheat seeds, and he got 3 or 4 different bacillus species from wheat, and then he sterilized wheat seeds, and then he set up an experiment in potting mix with and without microbes, and then measured the nutrients that went into the shoots of those microbes. Okay, and this is one of those results of one of those experiments, and the plants from that experiment. The one to the left is the control. This is with no bacteria. This is an azenic plant. So the plants grow, but their shoots are repressed. They're not as big, and the roots are somewhat repressed compared to when he used to sterilize plant, and then he put one of the bacillus species back on, and you can see the roots are now much bigger and the shoots are much bigger. And he did this with several of these bacteria, and this actually is this is analyzing the shoots for nitrogen, and you can see here the amount of nitrogen in shoots with no bacteria. This bar graph to the left, you see it's down here, and then you look at the bacterium Bacillus 1, that red Bacillus 1, that second, and it's about 30% higher again. For all those Bacillus species, it looks like it's about the same, about 30% higher, which is consistent with our nitrogen 15 experiment. Phosphorus, okay, it's very similar, but looks like it's about 20% higher when you put the bacilli there. You put your bacteria back on, 20% higher. Potassium, looks like for this second bacillus, or this bacillus number 1, the second bar, you can see it's almost 3 times as much as you have in the control. So potassium, it makes a huge difference. And it's different depending on what bacterium you use. So all bacteria aren't equal. You can see this bacillus was much better than the others for potassium. Calcium, you see it's also about 25% different, something like that, 25% increase. Sulfur, looks like a 35 or 40% increase. Manganese, you see it's a 35, 40% increase, a good increase. Magnesium, it's increased. So macro and micronutrients. Iron, any nutrient, boron, any nutrient that you can think of is impacted by the Rhizophagy cycle. Now, it may be that some are more important, like for example, potassium, and based on the previous experiment that we just looked at. The outcomes of the Rhizophagy cycle, there's beneficial outcomes of Rhizophagy cycle, and I'll just summarize them here. One is that because plants are doing Rhizophagy cycle, they're getting nutrients from soil microbes, so they have enhanced nutrient absorption. It's not the only way that plants get nutrients, but it's one way that plants are getting nutrients, and they're getting nutrients directly through oxidation of these microbes. And it may vary depending on which microbes are involved. It may vary depending on which plant. Okay, another benefit is that plants get increased oxidative stress tolerance. Okay, because these microbes are going in the plants and the plants are using reactive oxygen, superoxide to get nutrients out of them and to control these microbes in the plants. Because the plant is using that superoxide, that reactive oxygen, plants will upregulate oxidative stress genes, antioxidants, for example, and other molecules that protect it against its own reactive oxygen. And what that means is that plants are already resistant to other kinds of oxidative stresses, like drought, like heavy metals, like salt, like other stressors, heat, for example. And if you just feed plants with fertilizer, you don't get that effect. Plants are healthier when they're actually working for their nutrients. Another effect is that these microbes involved in the Rhizophagy cycle will actually go out into the soil and will alter the behavior of pathogenic microbes. They'll colonize these other microbes, like Fusarium, for example, and they'll extract nutrients from those microbes. And that weakens those microbes so that they're not able to cause disease. They don't have the oomph that it takes to actually cause disease in the plant. And in some cases, we can see those microbes that would be pathogenic, we can see them actually colonizing the plant, but without causing disease, and typically with bacteria around them and on the hyphae. So these are a couple of articles. They're open access articles about the Rhizophagy cycle. One of them in 2018 in Microorganisms. And if you just if you Google Rhizophagy cycle, you'll probably turn up this article and Microorganisms there. This article will pop up. It's free access. Another one in 2019 about endophytes, also talks about Rhizophagy cycle. This one is in actually Pest Management Science magazine. It's also open access. And many of my students are working on this process and have worked on this process and endophytes over the years. Visiting scientists, many of them are working on this. Collaborators have worked on this. This is not a one-man operation by any stretch of the imagination. Okay, and that is everything I have to share. And I think.
Wow. Holy crow, James. I mean, what a presentation. It just stimulates so many questions. Unbelievable. Can I ask a few?
Yes, of course.
All right. So let's start with the mycorrhizal fungi. How do these I'm going to back up for a second. I know these bacteria, the presence of these bacteria impact mycorrhizal fungi. I also know that mycorrhizal fungi impact root hair placement, and that the mycorrhizal fungi are capable of either putting out a signal themselves, or the plant does as a result of the response, to put a distance between root hairs, so that they separate from themselves for some reason. Could you talk a little bit about the interrelationship between mycorrhizal fungi and these particular bacteria and Rhizophagy? Yeah, yes. Okay, phagy.
Yes. Yeah, so the Rhizophagy cycle is a phenomenon that happens only at the tips of roots, only just behind those meristems and where the root hairs form, right? So a bit further up would be where you're going to get mycorrhizal, like for example, VA mycorrhizae, or even colonization of ectomycorrhizae. So in that case, they don't interact much at all. There would not be a big interaction between Rhizophagy cycle and VA mycorrhizae, for example.
Have you found mycorrhizal fungi in roots without the root hair, without the Rhizophagy going on? In other words, those.
Yes.
Okay.
It's a good question. Really a good question, Jeff. There are a few plants that tend not to produce root hairs. And like, for example, blueberries, right? Blueberries, and the blueberries typically are cultivated in very acid soils. And acid soils will inhibit bacteria. And so what happens in those plants, it really is interesting. What happens in those plants have adapted by affiliating with ericoid mycorrhizae instead of Rhizophagy. So they don't use these bacteria at all. Instead they replace them with ericoid mycorrhizae. And I have been told, I've not done the experiment yet. I know it blows your mind. There was an agriculturist, Greg Patterson. I don't know if you know him, but he's in A&L Canada Labs. He told me that he grew blueberries, and he grew it under high elevated pH, and he got root hair formation. So it really is in very, very low pH situations where likely Rhizophagy cycle just cannot occur. And so plants are forced to do other symbiosis like these ericoid symbiosis. It's really, we have not examined all the ramifications of that. You know, so I mean, as you might know, Jeff, more is unknown than is known, than is understood. I mean, you can see the kind of biology that we're doing, you know, we're seeing it, we're looking at it, that gives us certain capacity to, you know, to see what other people don't see, but we're also missing some stuff, you know, trying to figure out, you know, what are the genes that are turned on and blah, 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 all of that. That's all that it's so important, but it's more difficult to get into those aspects. Yeah, so thank you.
So, can I ask a question? Can you hear me?
Yes, I can hear you.
Okay, so
Yes.
how much do you think this affects the health of the plant as it goes on? I mean, I'm thinking that, you know, I come from a medical background.
Yeah.
And one of the things we've noticed is that a lot of the bacteria that infect people's guts that cause even serious disease, even after the person has been cured, it keeps a little colony of those microbes there for quite a while. And that seems to be important for the organism developing real resistance to those organisms. And, you know, I'm just thinking maybe there's a barrier between some of these microbes from the very tip going up, but they are developing the specific or rather specific immunity of the plant.
I mean, that's a really interesting question because you get into the idea of microbiome connected to plant health and organism health, eukaryote health. And we know, like you said, we know with humans, you know, we definitely have to have a healthy microbiome to be healthy, right? Well, it's the same with these plants, and you're absolutely right. These microbes don't stay in the root. They are moved throughout the plant. We know that for one reason, because some of these microbes end up on the seeds, and they're basically soil microbes that end up on the seeds. And we also, in some cases, we can see them in the shoot tissues of certain plants. And we did a big study with vanilla vine. This is a vine, vanilla pod, right? It's a vine plant. And it actually grows for periods of time in which it doesn't even connect to the ground. It can survive for just floating in the air, just in the trees, right? Arborescent, epiphytic kind of. But it carries its microbes on its shoot meristem. And you have a little community of microbes in the shoot of the vanilla vine. It's almost like a vase-like shoot. And the microbes are down in there, and the plant constantly keeps them moist. It's almost like a digestion chamber. But as that shoot grows, the tissues of the vine become infected. So the leaves, as they form, become infected internally, the cells internally, with these bacteria. And we don't know what would happen if those microbes were removed. But we do know that those microbes also do participate in soil activities. So when the plant, all of its tissues become filled with these bacteria, endospores really, because a lot of them are, one is bacillus, but when it produces its root, it goes down to the soil, then the bacteria are there, and it can then go and begin participating in the Rhizophagy cycle again. We do know that if we remove all the microbes from plants, they do not grow very well. I mean, if we grow these, I mean, you've seen the experiment. They grow. They're weak looking. I did an experiment during the COVID lockdown and with wheat, and the plants without the beneficial microbes died after with wheat plants. They died after like 3 months of unknown causes. You know, they're not. But when they had endophytes associated with them, they lived and thrived. So, you know, what is it? Why are they weak? I mean, I think it's probably oxidative stress tolerance, but it may be more than that. Plants don't like humans. We have like plants have most like plants and fungi, for example, will have this basic um, defensive system, fundamental defensive system, whereas humans have an more advanced defensive system. So some effects in humans may not be exactly equivalent. I can't say. I mean, that in terms of, is there really this immune development? I think it's mostly going to be an oxidative stress development and also direct defense response, because these microbes themselves are defensive for the plant.
Yeah.
But that,
Hey, Dr. James, since we're almost at the end of the hour, and I don't know whether we're going to end of the hour, if you're a cannabis grower, I can see right away certain activities that people engage in that they need to stop, and certain that they need to engage in more. The first one, I think, is, of course, this idea of using hydrogen peroxide in your seed germination process. Your thoughts?
Yeah, I think anything that that affects your microbes is not a good idea. It's not a good idea. And it turns out the hydrogen peroxide, I mean, what might you be doing there? You're not going to hurt the bacteria, the fungi. You're not going to hurt the fungi, but you will hurt your beneficial bacteria. And in fact, if some of the fungi are pathogenic, you're going to favor them over these bacteria that may help defend your plant from the fungi themselves. So,
Yeah, I can see a debate coming. That what you're doing is you're doing the same thing that happens inside the cell, and you're releasing nutrients out there on that little paper towel and feeding the plant root the wrong way, of course. But how about UV lights? What about the idea of using UV lights, lights with lots of UVB wavelengths in it? What does that do to the process?
So I cannot even answer that, Jeff. You've got me there. If you're now, for example, using UV to kill your soil microbes or your seed microbes, it's a bad idea. Anything that will hurt your microbes is a bad idea. Yeah, so I would not do that. Yeah, it may be a good idea to put microbes on there, would be my suggestion, or to put kind of compost tea or something that contains microbes to build up your soil so that they're more, so they have a better microbial diversity and higher microbial population. Maybe to use the microbiometer test to actually make sure you have lots of microbes there in those soils.
This is Laura. We have two questions from people. James, can you see the chat? I'm not going to pronounce the words properly. If you hit the chat button at the bottom.
Chat. Okay, let me see if I can. Okay, I see 18 things there. Okay, I see it.
If you go all the way to the bottom, there's a question second to the bottom from Jen Thomas. And Jen, if you want to unmute yourself, I'll do that too. Says, can you reiterate on what oxidative?
Yes, I certainly can. Yeah. So, I mean, you could think it's very similar to people, right? To how we have stress tolerance. What are we doing? We're trying to. Oftentimes, we're trying to take anthocyanins, or we're trying to take antioxidants to fortify us against that oxidative stress. But what happens in plants is we don't have those pills for plants. Instead, what happens is the plants, in degrading or exposing these microbes to reactive oxygen, superoxide, what happens is that the plants themselves need to upregulate or express at a higher level their antioxidants, their own antioxidants, and their other oxidative stress genes, like, for example, carotenoids, pigments that might be defensive against free radicals, right? And antioxidants, other kind of oxidants, superoxide and so forth. And that makes them more resistant against, for example, heat stress. Heat stress, besides the heat, it generates reactive oxygen, and that reactive oxygen damages the plant cells and weakens the plant cells and makes the plant so it's vulnerable to disease and everything else, and damage from the heat, anything else that comes along. Or salt, for example, you get a high salt in the soil, that also translates to more oxidative stress, more reactive oxygen that forms in cells and damages the cells and does damage to the plant. Okay, so the fact that these plants have these microbes and they're using reactive oxygen to control them and extract nutrients from them makes them more resistant to all these other kinds of stresses. Makes them hardier. They're hardier plants.
Thank you. And there's one more question. If you go up 3 or 4 more things, and I would say it, but I'm going to say the words wrong.
Okay. Which one is it? Which one is it?
Says Jim, when you compare the
yes.
root hair infection process?
Between Pseudomonas and Rhizobium, do you see any similarities or differences? That is really a cool question. I don't know who asked that
It's
question.
me, Prakash, Jim. Hi.
Prakash. Yeah. No, Prakash, that's a really cool question. Yeah. Yes. So Rhizobia. So I've known Prakash for many, many years. And, so Rhizobia, they actually will, at least they're believed to get into root cells by infecting the root hair itself from the outside. That's how they're believed to enter. And then they go in and they initiate tissue formation, which forms into this nodule structure. But, you know, I mean, there's good evidence that that's occurring, but there seems to be I mean, if we have this other process, this other means for these microbes to get in, to go in from the root tip, entering the root cells, and then colonize the root hairs themselves, that would seem a more logical way for those microbes to get into the rhizobia. Recently, it's been found that rhizobial nodules have a community of microbes in them. So it's not just a single species of rhizobia, which kind of favors this other means of entry. But I can't definitively say, because there's a lot of work that people have done tracking, you know, these little filament enter ways of entering where bacteria go into those hairs. So I can't say that that's not correct. I just have to say that there is this question in my mind, and maybe a few other people, that wonder, is that really correct? Or, you know, so that's it. So that's a great question.
Thank you.
Are these, excuse me, I'm so sorry. Are these hairs there because of this process? Is that why we have root hairs, so that maybe the plant is able to replace these bacteria out into a new area where they haven't already mined the nutrients?
Now, Jeff,
are they?
Didn't we all learn in third grade science class, that plant roots have hairs for absorbing nutrients from soil? Didn't we all learn that? That's what I learned. I don't know about you, but that's what I learned. But yeah, that's what we question. We question if that is the primary reason for hair development, because most roots don't have hairs. Most roots don't have hairs, only just right around that root tip where it's really young. And they seem to function so strictly for moving those microbes, those bacteria, out into the rhizosphere. And it's so logical, you know, well, why not just dump them right there on the root surface? Because the root surface has already been mined for nutrients. So putting them out there in the rhizosphere and then dumping them makes a lot more sense from this nutritional acquisition function. You know, and this farming, again, I go back to the farming. These plants are farming microbes, you know, from putting nutrients out. Yeah.
And we have a couple more questions.
Yeah.
One from Dr. Faust. What specific microbes seem the most promising? And also, could VAM, and I'm not sure which fungi that is, aid or reduce rhizosphere positive bacteria
Hmm.
in the roots. And Vic, I'll unmute you if you want to.
Okay, the first part I can
They're
answer.
in the chat. I think you can look at them.
Okay, okay. The first part I can answer. Which microbes are involved? You know, they're all the known ones that we know about. They're bacillus and they're pseudomonads, and we've gotten Acinetobacter, Burkholderia. We found some that are involved in, like Burkholderia, involved in Rhizobia. Even they do this. So I mean, they're common ones. We can cultivate them now. What is all in there? You know, it's going to take some metagenomics, I think, for us to really do a roll call of what are the bacteria that have entered. We know fungi also go in. So I didn't mention that. The yeasts, certain yeasts go in, and other fungi we don't know about. They form these little spherical structures also. Just mycologists have given them a different name. They call them mycosomes. Okay, now the mycorrhizal question, you know, I wish I had more explanation about that question. I don't know if I can see it in here. Dr. Faust, are you here? Was that your question? Can you come
Yes.
online
Yeah.
and explain it? Explain
Yeah.
that to me, and I'll try to
Well,
answer it.
I read a paper, you know, not long ago, and it was interesting because it was showing kind of a cooperation or a symbiosis between, you know, bacteria like Azotobacter and the microbial matrix. I mean, with a VAM with a VAM fungi. That seem to be beneficial on both ends, like classic.
I can address that now. I'm glad you told me. Okay, so there's two things. By the way, I'll tell you, I'm a plant pathologist and a botanist and a mycologist. So that's one of my favorite topics.
That's a very good combination.
Yeah. So I will tell you. Yeah, these bacteria, I mean, it's fascinating. They will also go inside your VAM fungi and other zygomycetes. Those fungi don't have cell walls, they don't have septa. And so some of these microbes can go in and they can just move inside the hyphae themselves, move around. And they'll colonize your VAM fungi in particular. And also the surface of these hyphae become like, I think there was a paper about it, but they essentially, we see it all the time. They become like superhighways for these bacteria. But they will go on the surface of these hyphae and then move around. So you can easily have, you know, VAM fungi going out into the soil being a conduit for these bacteria to move to the plant and out again.
Beautiful.
Now it's really cool. So, you know, plants do multiple symbiosis to get their nutrients. And of course, VAM fungi is one of them. Rhizobia, another. Rhizophagy Cycle is another. And just cultivating microbes in the soil, you know, causing them to solubilize nutrients. Just another thing that they're doing, you know, with their exudates. You know,
well, it's a nice way of saying it. You know, plants are farming.
Plants are farming. That's the brilliant thing. If we know that,
they're reaching and they're moving.
It changes, Dr. Faust, it changes how you think about plants. If you think that this is a process that they engage in, and you start things come together in a different way, changes how you think about plants, that they are actually in charge of this process.
Well, I think we need to get the point across to farmers.
I have a question on that. So this is Vic, and first of all, Dr. White, thank you. That was a fascinating presentation. And Laura and Julie and Dr. Jeff, thanks for inviting BioAct team. We get a few of us here. Appreciate it for the call. One of your comments fascinated me, that it's better for the plants to work, make them work to get the nutrients. I know that the VAM stop interacting, or actually the other way around, plants stop releasing exudate if there is high phosphate availability. Have you seen anything like that in the research where you provide ethylene to the plants and then they don't need to interact with the what's behind your comment that you got to make them work harder and it's better?
Well, they're working. What's behind that comment is if they have to get that microbe. Okay, here's the if you give the plant fertilizer, only fertilizer, those plants don't have to work for those nutrients. They don't need to degrade those microbes. They don't need to produce exudates and attract those microbes. You fed them with everything, okay? And those plants would not have to upregulate their antioxidative stress genes. So those plants would be less hardy than plants that are produced in a microbe-rich environment, working by oxidizing these microbes for their nutrients. And, that's something that that you could test for yourself in a simple experiments and verify. But that's what's behind that statement.
Got it. Yeah.
Would that be basically like you're stimulating the plant cellular metabolism, or?
In a sense, it is because they're using antioxidants, they're using reactive oxygen that they upregulate a lot of genes, and they've got to absorb the nutrients that they're getting from those microbes. That's causing metabolism stimulation. You are stimulating metabolism. I mean, we know with people, if you take all the microbes away from us, we become very weak. If we get all of our nutrients from IV, we're not going to be healthy. It's that feeding plants with fertilizer. Sorry, it's like IV.
Well, yeah, the other way to put it is these are the bottom of the soil food web. These organisms that you're dealing with, they're at the bottom of the soil food web. And if you replace them with chemicals so that the plant doesn't have to use the energy to get those exudates, so that the plant doesn't have to form a symbiotic relationship, which requires constant energy, if it can get away with it, not having that stuff, it drops all of those signals, all of those defenses, and it takes a while to get them back again. So that even if you're fertilizing all the time, there's always little gaps. Those don't exist when the plant is feeding itself naturally. It's always getting what it needs when it needs it, assuming you've got good soil. But as soon as you start screwing that system up by adding good stuff into the soil and making the plant go, I don't have to send that signal out to sit there with a nitrogen fixing bacteria. I'm going to get it for free. So much easier, but it's not constant and it's not as good.
Yeah.
Sorry.
It's true. We have one more question. Joanne? Johan, I'm sorry.
That's right. Not the first time, won't be the last. Yeah, I had a question. I from the kind of the other side of the coin when it comes to agriculture, being soilless agriculture or greenhouse agriculture. And until revisiting the soil over the years, I kind of now say the only thing missing from soilless agriculture is the soil, which would include our microbes and et cetera. I had a question about the microbial entry zone, but I kind of wanted to focus on this root hair topic, because when we think of greenhouse grown crops, typically up until recent years, there hasn't been any intentional inoculation of microbes. And I think Jeff mentioned, hydrogen peroxide, UV, et cetera. But yet in these systems, whether you're using something like rock wool or Oasis cubes, which is foam, you still see this proliferation of root hairs. So I'm wondering, has anyone done an endophyte count in plants grown in such systems that aren't purposely inoculated to see what species are getting into the system and to back up this idea that the root hairs and the microbes that development is from the microbial.
Yeah, no, Johan, that's a really good idea. No, we have not done that. And I think I have one of my graduate students was on this in the audience today, so maybe he'll consider that. But that's something that would be really interesting to do and to evaluate, what effect hydroponic systems have on Rhizophagy cycle, and can you maximize rhizophagy in a hydroponic system? It's possible, I mean, I would think that you could, and in essence, continue to make the plant work a little bit using rhizophagy. Just put the right microbes in there and put the right conditions. So, yeah, no, nobody has done that, and it really is a good idea to do that. Thank you.
But they have with mycorrhizal fungi, and you can grow mycorrhizal fungi in certain mediums. Rock wool, for example, you can grow the mycorrhizal fungi. It's not the easiest thing to do, but you can do it. You have to inoculate and maybe re-inoculate, etc. So if you can do it with that, probably ought to be able to.
Probably could. Well, the formation of the hair is an indication that they're doing Rhizophagy cycle. So they have some.
But would it make a difference in a hydroponic system when you're constantly bathing the root in nutrients?
Right, but yeah, exactly. But a rock wool system might be a little bit different if it's not totally soaked, right? If it's, yeah. So.
This is Laura. This is Laura. We've gone over our time, but no one seems to be moving out. So we could still do some questions for a while. I just wanted to say that this presentation and the PowerPoint, if it's permitted, will be on YouTube by Monday on the microBIOMETER website if you want to see it with contact information. And then I thought Judy might want to say something. She put the call together. And it's been really neat, especially for me. I'm new to soil science, so I'll turn it over to Judy real quick.
Okay, thank you so much, Dr. White. It's been so encouraging to read your work and to see all the credit you give to your students and how many enthusiastic students you have joining this. I'm sure that the understandings you're bringing are going to revolutionize how we go forward with agriculture and our appreciation of that. And as you've been talking, I've actually been just philosophically talk about the fact that children who are born by cesarean do not pick up their mother's bacteria as they're born. And they are much more susceptible, and their immune systems do not develop correctly. And so it's so interesting to me that this kind of newborn root, is tasting all the bacteria and things in its environment and developing this immunity that's going to help it, thrive and survive the actual experiences that plant is going to have in its environment. So I'd be really interested in following this up further in terms of, us understanding what is the interaction between how people are trying to put microbes down and the microbes that are actually in the environment and going to continue to dominate the environment of the soil that an agriculturist actually has to work with. And I don't, I think is, understanding that coming together, is an area where we need to help our farmer friends, the people we work for. I thank you so much.
You're so welcome, Judy.
Bye.
Bye-bye.
Bye-bye.
Thanks, Jim. Thank you.
You're so welcome, Prakash. Thank you. It was a great question. See you, Jeff. Y'all have a good night.
Good night, all.
I can see a book coming.